Why 5%? Where did that come from? Who ordered that? And it turns out that it came from the very beginning of modern statistical inference, which is Ronald Fisher's paper in 1926, focused on agricultural economics, that we should use this 5% as a method for determining whether or not you've got a scientific fact in that one context. But it was pretty clear from his statement that there were a number of other considerations in formulating that rule of thumb. But ever since then, we've used 5% as a standard across all fields, even those outside of agriculture. And it's gotten to the point where even the American Statistical Association, that venerated organization that all statisticians are part of, they've now made a statement that's been adopted by the entire organization of all statisticians, that p-value is not the only thing that matters. And there are two statements that the ASA makes of their 6 that I want to highlight. The first is that scientific conclusions and business or policy decisions should not be based only on whether a p-value passes a specific threshold. And second, a p-value or statistical significance does not measure the size or of an effect or the importance of a result. Size and importance. So I want to talk for a few minutes about measuring size and importance. Now, the trade-off that I just described in that LA Law episode is sometimes called the Blackstone ratio. Blackstone in 1765 said it is better that 10 guilty persons escape than that one innocent suffer. But it was Ben Franklin that came up with a 100-to-1 ratio. These statements are specific statements about how you trade off type 1 versus type 2 error. And, you know, they have different implications for things like the amount of crime that may occur and the amount of criminals that are on the loose. So, in fact, if we really do believe that it's better to let 100 guilty people go than to convict one innocent person, if we really believe that it should be 100-to-1, well, that suggests that there could be a lot of criminals floating around. Because if I were a criminal thinking about where I should practice my trade, I'd want to go to the 101 country versus a 10-to-1 country, right? So these things matter. And it matters for the FDA. In fact, even before the laws tell us that the FDA has to consider this, the FDA was already ahead of it in trying to think about how to incorporate patient preferences in this trade-off between type 1 and type 2 error. So in a paper that I co-authored with my former student, Vahid Monteza-Hojat, and a former pharma executive from Pfizer, Leah Isakov, we proposed to try to make this trade-off of type 1 versus type 2 error by measuring the burden of disease. And obviously, we were not experts in this, but there have been a number of studies published that try to capture burden of disease. So, for example, in the 2013 paper on the burden of disease from 2010, we actually have used measures of the years of loss due to disability, or YLDs, and ultimately various different data on prevalence and severity. And using this data, it turns out you can actually quantify severity in a very straightforward fashion. So here's a list of the top 10 causes of mortality in the United States as of 2010, and you can get a sense of how these methods work by looking at the severity column. Ischemic heart disease, 0.12. Doesn't matter what the numbers actually mean, but look at the differences in their value. Lung cancer, 0.45. Ischemic stroke, 0.15. All the way down to cirrhosis of the liver, 0.49. Alzheimer's disease, 0.18. You can actually capture from this very crude source of data a simple measure of burden of disease. Not necessarily the best measure, the right measure, but it is one measure. And based upon this, you can actually answer the question, what are the costs to society from the patient perspective of type 1 versus type 2 error, both after the trial, after you make your decision, but even during the trial. The fact that a trial takes 3 to 5 years, that imposes a cost on patients that are undergoing the trial, as well as those who are not receiving therapy because you've got to conduct the trial. It turns out that when you add all of these things up, when you take all of these costs into account, there is a framework for being able to compute the greatest good for the greatest number. And it's not a framework that we developed. It's a framework that was developed decades ago called Bayesian decision analysis. Basic idea is to minimize the expected loss from your decision, meaning you want to minimize the expected cost if you're wrong in one sense, false positive, as well as minimizing the cost of being wrong if you've got a false negative. And when you do that, you actually get numbers out. Instead of a p-value of 5%, you'll get potentially very different numbers. So we took this idea out for a spin by collaborating with a well-known biostatistician, Dan Sargent, who unfortunately passed away last year. But he collaborated with us on a particular project involving cancer. At the time, Dan was the chief biostatistician involved in the Alliance Cancer Trials, which is a group of a dozen or so cancer centers focusing on various different kinds of cancer. And Dan was generous enough to share with us his data for those clinical trials, and based upon burden of disease measures for different kinds of cancer, we're actually able to calibrate these kinds of optimization problems. And here are the outcomes for the Alliance trials that we looked at. There are a few things going on here, but I just want to point you to the red dots, because the red dots are the Bayesian decision analysis optimal trade-off between type 1 and type 2 error in terms of what the p-values are that would make that optimization work. And so you might not be able to see from where you are, but let me just point out one point, which is way up here in the upper left. This is the BDA optimal p-value for glioblastoma. And the scale is here on the right-hand side, and when you look over it, you see this is a p-value of 47.5%. Now, that is a ridiculously high number, and let me just interpret it for all of you, just in case you're not exactly sure what it means. 47.5% p-value means that the false positive rate is about half. Half of the approvals that you would be making in this case are duds, which sounds absurd unless you are a glioblastoma patient. Because as we know, glioblastoma is pretty much a death sentence. There are no therapies right now, and the survival rate over 3 to 5 years is single digits at this point. So if you have glioblastoma, you're out of luck. And for those patients, 50-50 seems pretty good. On the other hand, I want you to look at the other extreme down here. This is prostate cancer. And the p-value, according to this Bayesian decision analysis process, is 1%. 1%. It is much more conservative than the threshold that most of us are used to using, this 5%. Why is that? Well, the answer is pretty clear. First of all, prostate cancer affects a large population. Most men will get prostate cancer at some point in their lives. Sorry to tell you that, bad news. But the fact is that there are very good therapies for prostate cancer already. In many cases, it is not immediately fatal. And finally, because there's so many people that have it, if you blast out a drug or a therapy that's not effective, you are creating huge potential side effects across a large patient population. So you need to be much more conservative. So obviously, this depends upon the parameters of how you're measuring cost. And our use of the burden of disease measure is just one of many possible choices. But the focus that we have is not so much on any one number, but rather on the process by which we conclude that these kinds of p-values really have to be adjusted to different indications. So is this really practical? Well, in its current form, a p-value of 47.5% is clearly unrealistic. And part of the problem is a point that was made at an FDA presentation that we gave a couple of years ago. Once a drug is approved or a therapy is approved, it is very, very difficult to take it off the market. And that's the problem with a p-value of 47.5%. So what we're proposing is a completely new form of drug approval. And it's what, for lack of a better term, we call a right to try license. This new form of approval is a 2-year license to be able to market a drug in a very limited format with no off-label use and requiring the therapeutic manufacturer to engage in strict monitoring, data collection, and sharing during that 2-year period. So if you're putting out a device under this right to try license, you are obligated to collect data continuously on all of the individuals that are paying for this therapy and providing that data not only to the FDA, but to others as well. This license can be revoked at any time with any kind of adverse effect. And at the end of this 2-year right to try period, either the license expires or it converts to a regular approval as a function of FDA decision. And when you look at this, it looks a lot like an adaptive trial with one important difference. The difference is that drug manufacturers, device manufacturers, they can actually charge for this. Now, you might think that's a small difference, but it will make a huge difference in providing incentives for new therapies to be developed. If device and drug manufacturers can actually charge during this period, they'll have much greater incentive to be able to provide these kinds of therapeutics, to be able to develop them. So a 47.5% p-value, that may seem outrageous, but looked at in this context, it actually doesn't seem all that different from compassionate use in many cases and adaptive trials. So there are lots of qualifications about how this is going to work. How do you choose the parameters, the costs for the various different errors? Well, we believe that having stakeholders represented can actually come up with that calibration. Much like the way that in the oncology field, the FDA has organized this Oncologic Drug Advisory Committee. You can have an ODAC for these kinds of decisions, but now include patient advocate representatives. Whose preferences should be reflected? Well, that's kind of obvious. We think patients should be the focus. Potential backlash from toxicities and side effects. In this right to try context, I think that would minimize those kinds of issues, but they will still be there. And finally, there are going to be ethical considerations. But frankly, given the current system, we already have those ethical considerations. And having a more systematic approach to dealing with these issues would be far easier. To give you one case in point, I believe that the FDA would be the first to acknowledge that the process by which eteplirsen, the Duchenne muscular dystrophy drug that was approved last year, came about was not ideal. And that was a situation where there was tremendous amounts of pressure from the parents of DMD children. And it was very difficult to push back against that tide of public opinion, even though the statistical evidence for eteplirsen is, at best, weak. So let me conclude by giving you a preview of what you're going to be hearing about later on from other speakers, which is the wonderful collaboration that we've been privileged to participate with MDIC, the Michael J. Fox Foundation, and the FDA on applying these methods to Parkinson's disease therapeutics. In particular, the ability for us to collaborate with MDIC, RTI, and the Michael J. Fox Foundation allowed us for the very first time to collect data directly from patients and incorporate their preferences into how to calibrate the kind of optimal threshold for these kinds of decisions. And I won't say too much about this chart because I'm going to let Sho, who is the lead investigator at MIT, working with RTI, Michael J. Fox, MDIC, and FDA to do this. But the bottom line is that when you separate out the subgroups across their risk tolerance and their experience, what you see is that for certain parts of the patient population, 5% is way too aggressive, and for other parts of the population, it's way too conservative. Different patients look at these kinds of decisions in different ways. And so we think that there's great hope in taking these ideas and formulating them in a much more personalized way in order to achieve the greatest good for the greatest number. As the great biostatistician Don Berry put it, we should care about patient values as well as p-values. And thanks to all of you and the collaboration that we are so privileged to be a part of, we might be able to start doing that soon. Thank you.
Maybe have time for one or two questions, if you're up for any questions.
Thank you, Andrew. I really enjoyed your presentation. Unfortunately, I feel compelled to say something specific as a statistician. So one of the things was you were providing for the lay audience an interpretation of what p-value 0.47 means, right? And so the way you described it, it was you're saying about half the drugs that are approved are done, then emphasize, and half of them actually work.
Right.
A p-value is actually computed under the assumption of the null hypothesis, which is to say nothing works.
Right.
Not on the assumption that half of them work. And so the p-value is actually set up to say, in a universe where there are no effects, how often do I falsely assume some effects exist? That's where you get to 0.47. Not from saying that in a universe of drugs, how many would we then push forward? So it's the kind of difference that I think underscores why they came out with their statement on p-values, which is to say that you can be very easily misled if you do not consider the broader context. That's why they also have 10 statements, not just one, but a couple.
Right.
But I very much do support what you have gone through, which is to provide additional context for how we would incorporate other aspects of the testing procedure, such as patient preference, to then calibrate where you might make that decision in different groups.
Right.
So I encourage you to do that, but we need to be very cautious as we go forward that in the process of simplification, explaining this to a non-technical audience, we don't actually lead them astray. And that's part of the reason why I bring this up here. So
right.
Caution, please, for that.
Yeah, thank you. Point well taken. And by the way, I should also add that, you know, the point that I made about taking other considerations into account, that's not to say that the FDA doesn't do that now. In fact, they do that now. Very clearly, when you talk to CDRH, they take all sorts of considerations into account. The real benefit of this framework is that it can be done in a more systematic, observable, repeatable, transparent framework. But yeah, thank you for that clarification. I agree.
Yeah, I think it's probably a similar point, but I just want to be maybe more explicit for the non-statisticians. Right? So I think it's really important that we all recognize that a p-value of 0.05 is not the same thing as a type 1 error rate of 5%. Right? That's a posterior probability. And a p-value of 0.05, depending on your prior, can correspond to a type 1 error rate much lower than 5% or much greater than 5%. And so hopefully, somewhere in the math here, as we go through the presentation, we're going to be really clear on whether we're dealing with Bayesian posterior probabilities as the inputs to this model or plain old p-values. And if it's plain old p-values, how do we get from those to what actually is the type 1 error rate in our trial?
Right. And actually, let me add to that, which is that this framework that we're using, which is called Bayesian decision analysis, a number of true Bayesians would look at this and say, no, there's not very much Bayesian about it. From our perspective, that's a feature, not a bug necessarily, because I think even today, many people in the medical profession are not necessarily as familiar with the Bayesian approach. We have a paper that we're working on right now that uses this Bayesian adaptive trial framework and extends that framework to incorporate these kinds of considerations. So very much along the lines that you described.
Thanks very much for a very nice presentation. I'm Matt Maltese from Children's Hospital in Philadelphia. Your train analogy has really got me thinking, particularly the one with the two buttons and the switch, where you have one person on one side and 6 on the other. And I've also had the benefit of working both in safety at Department of Transportation and in medical devices in connection with the FDA. And so the human trade-off happens all the time, particularly when we're talking about funding and decisions and things like that. So the ethical question that I'd like to, maybe I pose it, you don't have to answer it, but what happens when the one is a kid?
Mm-hmm.
And the trade-off becomes, do I sacrifice a child versus sacrificing 6 adults? And I pose that because in terms of the resources we put at medical devices in particular, but other things, including in the safety world for children, there's a tremendous financial imbalance there. So I'll leave it at that.
I couldn't agree with you more, and you're absolutely right that in that ethical dilemma, if we actually change that one individual to a child, it definitely does make a difference. And it is odd that despite the fact that all of us agree that children should be a priority, that when it comes to pediatric devices, we're just farther away than for adult devices. I didn't understand that at first until I started looking into the economics of the situation, and it just turns out that the economic incentives aren't there. They're not as great for pediatric devices, and so I think we need to do something about that. But what we need to do is a little bit less clear. Policy, I think, could play a very critical role. Incentives along the Orphan Disease Act of 1983. So pediatric conditions are often considered orphan diseases, so there may be a way to deal with that, but I don't have the answers. Great, thank you.
All right, well,